So, oh, there's the chart. I'll start. Is this working? So hi, my name's Doc. I'm going to present some joint work with Jeremy Berg and Graham Coop. And in this work, what we're trying to do is to use sibling-based effect sizes to understand stratification and standard GWAS, so that we can then use that to understand selection. Those of us who are here in this room are probably here because we care about natural selection driving different populations apart on some quantitative trait. One of the standard frameworks for looking at this statistically is the QST-FST framework, which comes from the early 90s, from this pair of papers looking at Daphnia and Drosophila bizzotti. And what you do in this framework is you take a set of populations, you look at how genetically differentiated they are, you compare that with how differentiated they are on a quantitative trait, and you compare it to a null distribution. And that null distribution is generated by noting that under neutrality, the allele frequency difference between a population at some locus is going to be uncorrelated with that locus's effect on a quantitative trait. And once you know this, you can start building null distributions and doing hypothesis tests. So I work in this horrible, awful model system, Homo sapiens, and we can't do the common Godwin experiments you normally need to do the QST, FST kind of work. So there's a lot of excitement about doing this kind of thing with polygenic scores instead of actual quantitative traits. And so to get a polygenic score, what you do is you conduct a genome-wide association study, or a GWAS. So you're just regressing genotype and phenotype. The phenotype values on the y-axis here, the genotypes on the x-axis. You get a slope from that, and then you weight the individual's genotypes by those slopes. You're just taking a weighted sum of an individual's genotypes, where the weights are the effect sizes. That gives you the polygenic score. So it's just a prediction, an additive prediction of an individual's trait value on the basis of their genotype and some GWAS information. So using this kind of approach, there's been a lot of papers looking at evidence of natural selection on height within Europe. This is a paper from my co-authors about 5 years ago, suggesting that polygenic scores for height within Northern Europe are higher than in Southern Europe. There's been a lot of other papers doing this over the past 5 years, using different but similar statistical methods and pulling in their GWAS effect sizes from different places. So the key assumption of this work, remember in the QST-FST framework, the key assumption is that allele frequency differentiation and effect size are going to be independent. And in the polygenic score framework, it's the same but a slightly different assumption. It's that the estimated effect size and the allele frequency change are independent. And under selection is one reason that these would no longer be independent, right? But another reason is population stratification. Where, for those of you who don't speak GWASese, that just means you've got a set of individuals where you're doing your GWAS. There's some degree of population structure within that set of individuals. That structure is correlated with the phenotype, and so any SNPs that tag that population structure, that ancestry, end up looking spuriously associated with the trait. And there's a whole industry of correcting for this, but as we'll see, corrections have to be really, really perfect for these polygenic scores to work out. So there's been some renewed attention to this recently. A couple of papers came out in eLife earlier this year revisiting these height results. So one of these papers, they're just replicating the original result using effect sizes from this study called GIANT, which was the original GWAS for height. What I'm showing you here is latitude on the x-axis and height along the y-axis. I've always wanted to do one of these with some personality traits, so it could be changes in latitudes, changes in attitudes, but I've never done it. So if you do it in GIANT, you get this result. If you do it in the SIBs, so this is a different way of getting the effect sizes that I'll talk about in a minute. If you do it in a different study, the result goes away, or is at least much reduced. And I should say, this is true even though the individual GIANT effect sizes are not badly biased individually. So this is not to trash GIANT as a GWAS. It was a quite successful GWAS. The effect sizes are close to right. The big hits in GWAS are the big hits everywhere else. But if you have small stratification errors, they can really stack up into a polygenic score. Another way of looking at this is instead of thinking about population level or individual level polygenic scores, you can think about locus level effect sizes and their loadings on different principal components. And if you do that, these are the correlations in one PCA between giant effect sizes and various PC loadings. So you can see pretty big correlations on a couple of these that could be due to selection in principle, but we see that if you do this in a different set of data, in this case the UK Biobank set, those correlations tend to go away, suggesting stratification. So what I'm doing is jumping off from observations like this and trying to ask questions about can we build a statistical framework that will let us formally test for population stratification in a set of effect sizes? And can we do things like identifying the proportion of variance in a polygenic score that's attributable to stratification? So the tool we're going to use to do this is sibling-based effect sizes. So I'm just showing you a little pedigree. We've got a pair of parents, and they've got two daughters here. Mom is a heterozygote at this locus that I'm tracking. And the difference between these two siblings, these two sisters, is that they've gotten different alleles from mom. So meiosis happened a little bit differently. They grow up, they have some phenotype difference. We come along, we measure it, and then we correlate that phenotype difference with the genotype difference. And the real usefulness of that is that sibling differences in genotype are going to be due to this randomness in meiosis, so they're not going to be confounded by environment or by unlinked genetic loci. They might be mediated, the effects might be mediated environmentally, but they're not going to be confounded by environment. So once we have these effect sizes, what are we actually going to do? Well, we're going to build polygenic scores using standard GWAS and using SIB-GWAS. And we're going to compare the variance in those polygenic scores that lies along different principal components of the genotype matrix. There's two reasons to do this. One reason is that the PCs of the genotype matrix are a sort of fundamental way of representing population structure. But the other reason is that we can sort of change our point of view in an interesting and useful way that gives us the same results. And what we're going to do actually is instead think about instead of thinking of the individual level polygenic scores and the individual level coordinates in PC space, we're going to think about the effect size. And in particular, what we do is this. We can do exactly the same variance breakdown along the different principal components by summing up the eigenvalues so this is the eigenvalue of the jth PC multiplied by the dot product of the loadings with that PC times the effect sizes. So you can think of it squared. You can think of that dot product as being like a covariance between the effect sizes and the PC loadings. And this gives you exactly the same proportion of variance along the principal components. Once you do that, and once you think about effect sizes, we can then think about modeling the effect sizes. So our model is going to be very simple. We're going to think of the GWAS effect sizes as being the sum of some direct effect plus some stratification bias and some measurement error. Whereas the sibling-based effect sizes, I've argued, are robust to stratification, so we'll think of them as just the direct effect plus an error, a different error. Once we have this, we can start doing estimation and hypothesis testing by plugging it into that previous variance breakdown. I'm going to do this with 1,000 genomes samples from Europe. These are about 500 people from across Europe. I'm going to use effect sizes for height from the giant study and from a set of siblings from UK Biobank, and ask whether the stratification is significant and how much of the variance can be explained by stratification. So I'm showing you now results for a polygenic score where we're using all the SNPs in the genome after doing some clumping procedure. So what I'm showing you is proportion of variance on the y-axis along various principal components. These orange dots are the siblings. So you're probably wondering, why did you choose this terrible axis where I can't see anything? And the reason is I need that space to show you all the variance in giant that's along the second principal component. It's about 70% of the variance in this polygenic score. It lies along this north-south principal component within Europe. So that's highly significant. I probably didn't need to come up with the hypothesis test, but there it is. And when we plug this into our estimators, this suggests that about 60% of the variance in this polygenic score is due to stratification. So even though these individual errors are very small, they're driving a lot of the variation in this polygenic score. So I just want to say, there's not just one answer to this question. This is when we use all the SNPs. If you limit to a p-value, ones with p-value less than 0.01, you see a similar picture, but the answer changes in terms of the actual proportion of variance. So in this case, it looks like more like 25% of the variance ends up being due to stratification. But it's still quite substantial, right? I also wanted to mention quickly, it's not going to just be height. I don't have sibling effect sizes for this yet, but here's BMI. And I'm showing you now UK Biobank, and we still see that giant looks quite unusual compared to this other GWAS that we think is less stratified. And it's not going to just be giant either, right? We're going to be looking at a lot of different sets of effect sizes for doing this. Presumably we'll find stratification in other places as well. So a couple of takeaways. I hope I've convinced you that comparing sibling-based and standard GWAS effect sizes will inform us about stratification. And also that stratification, even if it's very subtle at the per locus level, can really explain a lot of variance in a polygenic score. That's important not just for us when we want to look at selection on polygenic scores, but people want to use polygenic scores for all kinds of things, other research purposes and even policy purposes. So it's important to understand this stuff. Let me just quickly plug, I'm starting a lab at USC in a year. If you know anyone who wants to be in LA for a postdoc or PhD, please talk to me. I'm also hawking a book. This is a statistics book that's at the OUP table. I'd love to talk more with you about it. So thanks to the Coop Lab for discussion and NIH for funding.
Yeah, I'm just wondering, did you assume additivity? I'm just wondering how well you estimate effect sizes like heterozygous versus homozygous.
Yeah, everything's assuming additivity. Yeah.
So why do you think PC2 is important? What is that separating out in biobank?
Oh yeah, so yeah, good point. That's PC2 here. The question is, why is PC2 important? PC2 is so that's PCA within the 1,000 gene European set, and it's the north-south axis essentially.
If you see differences between, so if you're comparing siblings produced by one heterozygous parent and one homozygous parent, and comparing the homozygous for either of the two alleles, do you see differences in the effect size in the siblings in those two cases? What do you infer? That is, if you see non-additivity.
So if you look if you have two heterozygous parents,
you have a heterozygous parent and a homozygous parent. A homozygous parent can be homozygous for either allele. So then you can compare a heterozygous offspring and homozygous. It's a homozygous sibling, but that sibling can be homozygous for either of the two alleles.
Right.
In those two cases. You can see a difference in the effect size of the allele. What would you infer from that?
Yeah, so generally people assume an additive model where it's going to be the same, so they're just pooling every sibling for that when they do these estimates. I haven't thought through what you would think if you did that comparison.
I was wondering if you could use this to estimate the covariance between the stratification bias and the true genetic effects across either within populations or across a landscape.